So one of the projects that I'm really interested in currently doing at the moment is investigating why a specific risk gene may influence our everyday metabolism and downstream influence our chance of developing the disease. So the gene is known as ABCA7, and individuals who carry a mutation in this gene have a 20% higher chance of developing Alzheimer's disease as they get older. I'm Dr Luke Whiley and I work at the Curtin Medical Research Institute and the School of Diagnostic and Therapeutic Sciences. I started my studies at the University of Surrey over in the UK, and following that I then moved into London. I went to King's College London, where I studied a PhD in translational medicine, studying Alzheimer's disease and trying to identify biomarkers of Alzheimer's disease. I then took some positions in industry, in analytical chemistry, trying to advance my skill set in analytical chemistry, in particular a technique known as mass spectrometry, before returning to a postdoc at Imperial College London, first at the UK National Phenome Centre at Imperial College London, and then a second postdoc at the UK Dementia Research Institute, again also based at Imperial in London. Following that, I was then headhunted to come across to set up a phenome centre, the Australian National Phenome Centre, based in Perth at Murdoch University, where I came across to help set the laboratory up, brand new operational laboratory. And more recently, back in July, I took up a position here at Curtin University. I specialise in the platform known as metabolic phenotyping. So we know that the genetics are the building blocks of who makes us what we are, but that's not really all of us. We also interact with the world around us, our environment, the dietary habits we make and interact with, the lifestyle choices that we make, and they all come together to really make us who we are. And I use a technology platform known as mass spectrometry, where we measure the metabolism of an individual, particularly across population health studies, and apply that to try and find out who and when people are likely to develop diseases, and try and predict their health outcomes that they're going to encounter in future. I use this platform specifically an interest to study diseases of aging. So dementia, Alzheimer's disease in particular, are something that's passionate to me, and I apply these platforms to try and identify who is going to be particularly at risk of developing Alzheimer's disease as they get older, and why they're going to be at risk of developing the disease. We know that there are genetic risk factors that can make people have a high chance of developing Alzheimer's disease. These have been identified in large-scale genome-wide association studies from large populations, and we've identified risk genes that can elevate an individual's chance of getting the disease. But what we don't know is why those genetic markers actually increase a person's chance of developing Alzheimer's as they get older. So one of the projects that I'm really interested in currently doing at the moment is investigating why a specific risk gene may influence our everyday metabolism and downstream influence our chance of developing the disease. So the gene is known as ABCA7, and individuals who carry a mutation in this gene have a 20% higher chance of developing Alzheimer's disease as they get older. But what we don't know is why that is, and even more, we don't actually know what the variation in that gene does to a person's everyday biology. They don't experience any developmental changes. People live healthy lives. It's only as they get older they have a higher chance of Alzheimer's disease. So what I'm doing, I'm using these mass spectrometry phenotyping platforms. I'm taking blood measurements of people's metabolism from a large population study based here in West Australia. That's the Butterton Population Medical Research Institute and their Healthy Aging study. I take genomic data and blood plasma metabolic data, and I try and correlate those to see if there's any differences in people who carry the risk genes to people who don't carry the risk genes. And it's like building a jigsaw puzzle. If I can put the pieces in place to see how those genetic risk variants are changing someone's metabolism before they get the disease, eventually I hope to build the picture together to say why they may then go on to develop Alzheimer's disease as they get older. This project collaborates across Western Australia, so first of all with the Boston Population Medical Research Institute, also the University of Western Australia, and the work itself commenced over when I was doing my postdoc in London with Imperial College London and the UK Dementia Research Institute. This is where we first identified the relationship between risk genes and variants in these genes, two aspects of our metabolism, on our everyday metabolism, looking at some cohort data over there. The work since expanded, and I've received fellowship funding from Dementia Australia and the Roy Simmons Foundation, who gave me their mid-career research fellowship to continue this work and to look into a population, an Australian population. The advantage of the work that I'm looking at now is that the population is longitudinal. So I've got sample time points throughout people's aging, so collected 6 years apart. So I can see how these relationships between genetics and metabolism are changing as people get older and move towards an age where Alzheimer's disease becomes more prevalent. My grandfather lived with dementia. He lived with vascular dementia, and I saw firsthand not only how the disease affected him and changed him, but also how it affected those around him. So my grandmother, my parents, our family, and the impact that disease had on his life as it progressed and affected his cognition and who he was. That was my initial experience of dementia, and it fascinated me that these diseases can have such a large effect on us as individuals, and yet relatively we know quite little about why they start and who they're most likely to affect. Without knowing that information, it then becomes very tricky to identify diseases that may modify an individual's chance of developing the disease. If we can develop interventions that not necessarily cure the disease, but even delay the onset of these diseases, it can make a huge difference to people's lives, not only on an individual basis, but those of their friends and family around them as well. So that really drove me to try and find out more about these diseases. That's where I started my PhD, where we were trying to find predictive diagnostic biomarkers for Alzheimer's disease and dementias, but then progressing on further to go and work at the UK Dementia Research Institute to try and find more about the mechanisms and understanding of these diseases and identify the risk of these diseases and who's going to be more chance of developing them as they grow older and go towards the ages where the dementias become more prevalent in our society. So outside of work, I'm kept relatively busy. I've got 3 young children, 3 under 5, so they keep me pretty busy outside of work these days. And when I get a small window of time, opportunity to go out and enjoy myself, I like to go running or cycling. I've got a deep interest in cricket as well. I played a lot of cricket, and I still try and play the odd game where I can when the family allow. But I really like sports, getting outside. Living in West Australia gives lots of opportunities to get outside with the family, and I'm also a big advocate of the park run community events that happen every Saturday morning. We go down and we run around the 5 kilometres, and so yeah, getting outside, running, cycling, sport, cricket when I can. So yeah, really like to go out and about.